So today, what I wanted to talk about here just was the biology of natural farming and kind of explain a little bit of how I'm using the microscope to verify and validate the different things that we observe. Like you see the plant grow really well. I'm trying to use the microscope to make that correlation where you can visually see it and test and see what biology is making plants grow better. So what you need to do this kind of analysis in soil is a microscope. This one here is 400 times, so it can zoom in at least 400 times. It also has this light down at the bottom here. It's this, this is a halogen or like LED light down there that's really bright. It goes through a condenser, and then it comes up. And those are the components that you need enough high power light to see the organisms because they're very small. We're looking at 1 micrometer in width, which is very, very tiny. It's 1,000th of a meter, or maybe
10,000.
million.
Yeah, it's pretty small.
It's really tiny, whatever a micro is. These microscopes here, it runs about $300 to $400. The other microscope I have, like I just got these microscopes here for the school, and this was about 300, and then the camera was another 50 onto it. So when you're looking for a microscope, another nice thing to have too is that if you see here when I'm twisting this, the slide moves, like the slide is moving around, and so it's real easy to then track and find the organisms.
Mm-hmm. Did you say where you got them at?
Microscope.net is where I went online.
Okay.
And I also have those microscopes at the school garden too, so if you're wanting to use them, maybe make an exchange to do that. So after I have my microscope, the next thing I wanted to go over is preparing a slide.
The connection between the, maybe she might have a connection with that. What you looking for?
Well, the thing is, it's going from this, and I have.
Get the connection between the.
Okay, let me go check.
This is
Oh, cool. So what I have to prepare slides is in this bag here, and I use this medicine dropper. I got this at Long's. It's just a thing that measures in milliliters here, and the milliliters are important to me because what I want to do is to dilute my slide, typically 1 to 5 or 1 to 10, and so. A cup of water. Okay, perfect.
Cross-contamination.
Yeah, I know, we're gonna. So what I'm gonna do here is to prepare my slide, and I want to be somewhat scientific with this and be consistent across my thing, is if I prepare slides differently, I'll get kind of different results. But this is a pretty consistent way to prepare slides that I'm going to share with you. And I have 3 different samples here. One is from an IMO pile, this one. This one is from my garden outside, and this one is from another part of my garden. where they're all looking really healthy. So I try to pick some healthy soil samples, and notice how this one made the bag already kind of like wet. So I know that whatever's in here is alive because it's breathing. So I'm just gonna open this bag and use this as my soil sample here to prepare the slide. So I want to do it 1 to 5. So what I do is I fill up my measure to 4 milliliters of water. And so it's at 4 milliliters of water, and then I add in dirt until it goes up to 5. And then I know I got enough 1 to 5 in there.
Isn't that 1 to 4?
Not the way I understand it.
They count it a little different.
I guess so. I thought 4 parts of one and 1 part of another. Be 4 to 1, 1 to 4.
This is the way I'm consistent across.
Okay, yeah, that's the important part. Yeah.
Yeah.
But you can say it's 1 part in 5.
1 part in 5. Yeah.
I guess mine could be slightly stronger than I don't know.
As long as you do it consistent, it doesn't matter.
1/5.
So, yeah, and also you change your dilution based on how your sample ends up looking. You know, like if I'm looking through and I can't see anything, it's too muddy, then I can dilute more. If I see, I'm trying to count certain things. But 1 to 5 I find is good. So you see now it's up to 5. I got my thing. So then the next part is to shake it because all the biology is on this stuff. And so just as consistently as possible for about 30 seconds of just, I'm not trying to like super tear it off. I'm just trying to be pretty even with this. And then once I'm satisfied with that, I take just a drop. Drop that onto my slide. And you can do either 1 or 2 drops, depending, just be consistent with it. And then take my cover slip and put that on the top. And then I kind of, see how it's like all a cluster in the center? I kind of like rub it around a little bit just to spread the water out a little bit. Try as best as possible to make it big. So you can see kind of, I got like almost over half the slide here. Probably be better, there's a clump of dirt in it though that's keeping it from totally going on there. Spread that out. Actually, I probably should spread it out better. So that's how you can make a slide. I'll have some slides over here, so after we're done with this, you can make slides and use the microscope to check it out. But 1 to 5, shake it 30 seconds, take a drop. So this next slide here says biology in a 600 to 600 soil sample, which what that means is the biology where I have 600 micrograms of bacteria and 600 micrograms of fungus in a sample per gram, I guess. And that's the ideal conditions that we want for most of the crops we're growing. So if we're growing taro or sweet potatoes or cassava or things that are crops like trees, coffee trees, those types of things. That's the range we want to be in. And so what I have written here is what you should see on a slide if I'm in that 600 to 600, if I'm balanced with my things, what I'll see. And on here I have 1 nematode per slide. So what I do is I put the microscope to 100 times power, and then I just scroll across the slide. nematodes, you're looking, once you find the nematode, you can identify what it is. But for every slide, I should have one nematode. I should have about 100 flagellates on there, which are bigger life forms that have a flagellum on them, and they just kind of bumble around in the soil, and they help to cycle nitrogen and cycle nutrients throughout the system. You shouldn't see a lot of ciliates, which are attracted when oxygen gets cut off. And every view on here, I should see a beneficial fungus. So for every slide, I should see one nematode, but for every place I move the microscope and move that slide around, I should see one fungus. So there's a lot of different funguses I should see on this slide for it to be in my in the balance thing. And I should also see some active bacteria. A lot of times I look at the thing and it's just all dead and nothing's moving around. But in a healthy soil, I should see some things wiggling around and moving and interacting. So those are the main things I have here. So what I'm going to do right now is just show you some pictures of the guys that we're looking at, and then we'll put this live through here to look at what we have. So in the IMO and in soil samples, we're looking at about 8 different creatures and life forms in there. The first thing you'll see is a lot of bacteria. And they just kind of look like chunks of dirt. They're kind of glued together. They may be round, they may be stick-shaped, but they're really, really tiny, and they're all clumped together. And you'll always find bacteria. Everywhere you go, you'll find bacteria. Then the next level up from that is an actinobacteria, which this is where this form started to turn a little bit more elongated and into tubes. And this is on the edge of going from, like if you see a lot of this in your sample, it's starting to go anaerobic. It's starting to run out of oxygen. This is also responsible for the smell when it rains. The smell that comes up is actinobacteria. There's tons of them everywhere.
How does your soil run out of oxygen?
Compaction. So these are the guys that restore oxygen back to the soil, and this is what I'm most focused on is beneficial fungus. And when I'm looking through the microscope, that's what I'm trying to see to determine the health of my soil, determine if my inputs are working. So beneficial fungus are parallel tubes in the soil, and they're darker brown in color. They tend to be, like parasitic fungus are clear, whereas beneficial fungus are darker colored. And they're also wider than 3.5 micrometers, so they tend to be wide in the soil. They may break into chunks, but they'll always be really nice and wide. And so do you see this fungus here, this picture, that long tube? That is the chunk of fungus. And during the process of shaking it up, you break them all into pieces. And so that's just inevitable that you'll find them kind of broken up in there. Yeah. And then the other really exciting thing that you'll see, and this is another good measure of soil health, that Kunhu Wei, I think that's how you say her name. She studies these nematodes specifically, because these you can see at 100 times power. And they tell you a lot about the soil, but they're little miniature snakes. And so they'll be like swimming around these snakes. And a lot of times people associate nematodes with bad guys, but only typically root feeders are bad for farmers. And these guys are everywhere. So having good, healthy nematode populations keep your root feeders in check. So a few other things you may see in the soil samples are things that are budded in a row like this. And these are yeasts, which a yeast buds, and they're anaerobic fungus. So they're living without oxygen, they're fungus. That's what makes beers and wines is yeast.
So that's not a good thing to see.
It depends, not in large quantities. If you're making like a compost tea and you see a lot of yeast, you know you've lost oxygen and you're probably not growing what you want. This is spirilla, or spirilla, or I forget the exact name, but it's named because it's this spring-looking thing. And what this is a pathogen. It's associated with disease. Like if you eat a whole lot of this, you'll get food poisoning. And that's why it's always good to have the microscope to check what you're working with, because this is present in an IMO sample that I checked out and had this spirilla in there. So, you know, you're putting out good microbes, but you may also put out these guys. So verifying, looking quickly under the microscope, you can see that, you know, what is it, I don't know. And then, I don't know if you can see it here, but it's kind of like Godzilla looking thing. It's huge on there. And what this is what they call an arthropod, which is kind of like a beetle or like a mite or like a thing that crawls around in the soil. And so it looks huge when you're zoomed in so much. Sometimes you'll find their legs and stuff broken off, their antennas, lots in the soil, especially compost piles. And then this is just what IMO 4 looks like, that when you put IMO 4, the sugar and the osmotic pressure makes the things form like globs and spores, and there's not much active stuff happening in the actual input. So if you just take the inputs and look at them, they're not that exciting. They kind of look like this. But once you dilute them and you get them wet and you make the sugar not so powerful, then this will sprout into life. So it looks, but every once in a while you will find some fungus living in IMO itself. And this is the basic theory of why you would use the microscope right here, is that if you take a soil sample and you look and you only find bacteria, that really only harbors weeds. So the plants that live bacterial oriented have weeds. When you start the soil sample and you start to change your fungus and bacteria ratio, the plants that start to grow, fungus really likes trees and slow growing, woody, lignin things. So it's on this extreme. Whereas bacteria is really fast growing, quick turnover, and so it tends to favor weeds, which just flower really fast, die, cycle those nutrients really fast. Whereas a tree is really methodical and slower. So as you start to shift your ratios out to become much more fungal, then naturally weeds won't want to sprout.
I'm looking forward to that day.
Yeah. Yeah. And so yeah, and using the microscope to be able to tell you when you do an application how effective it was, and so
So what do you want in between?
in between.
In between fungus and bacteria? What's the median?
About 600.
Yeah, equal parts by weight.
And that's where the vegetables grow best, is when the bacteria and the fungal match.
Yeah, certain veggies like lettuce, for instance, is not gonna be as hooked into fungal. Like the fungus will unlock nutrients in the soil, but the fungus isn't gonna actually plug into the lettuce. Whereas a tree is actually gonna plug into the fungus,
Okay.
as well as the fungus out there dissolving rock into nutrients. So certain plants really associate really heavily with the fungus, and other plants,
Do the bacteria dissolve rock too?
probably, but fungus is like, that's what it does.
That's its main purpose, yeah.
Yeah.
Huh.
Give the give it.
This sounds like I'm borrowing. One of the other agents is still here.
So, let me see.
What magnification do you have right now?
What power are you on?
Right now, I'm looking at it at 100 times.
Okay.
So, so this sample is pretty dirty. Like, it didn't aggregate very well. But this, so I was looking for some nematodes, but I found this right here, which. So I'm trying to like balance this whole.
Oh.
So what is that?
Yeast?
Yeast.
Yeah.
It's a yeast, right? You see how it's
It's
this
budded.
is budding. They're budding on each other. These two. Those are yeast right there, and then that might be the fungus. So and then this right here, this is a really nice example of a nice beneficial fungus. See this dark color, this uniform shape here all the way through.
Hey, Joe, you see the fungus is a little black thing moving around? Yeah.
Yeah.
You know what that is?
So that you know, this is actually kind of perfect. You see how that's one bacteria. So do you see the size of that black dot on there? I'm going to assume that that's 1 micrometer in length, because bacteria are typically about that size, 1 micrometer in length. And so you see how it's dancing around out there. You could take maybe 3 or 4 of those and put them side by side across here, like 1, 2, 3, 4, and that would be the width of this fungus, right? So you can see how 4 of those would fit across. So that gives me a scale to know that this fungus is at least greater than 3.5 micrometers. So I would say that this is definitely a beneficial fungus. So you use this tiny, like 1 bacteria to get your scale to see, and then you measure like, okay, so that it's wide enough, right? If it was only half that size, I would say, oh, that's not a beneficial one, because I can see that all the way across. So, and then sometimes here, Can you see what I'm doing? I'm scrolling in and out. I'm zooming in and out. And when you find a fungus, you want to kind of zoom across it to make sure that it is parallel the whole way, the edges are parallel the whole way, because then you know it's actually a fungus and it's not just like a antenna or like a leg or something else. They tend to be more round. So what I try to do is just scroll to some random spot and then look to see what I'm working with here. And so you guys can't see it. I have a little bit wider field of vision here. But this guy was in my field of vision. So I scrolled to some random spot on my slide. I just went to like a random view is what this is called. And in this view, I found another fungus. So in my view, found a fungus. So I know that I'm getting closer to that equal ratio. Because everywhere I go, I should find at least one.
This thing's moving.
So
See moving.
try to just go to some random spot.
This was your soil sample, not your IMO?
Yeah, no, this is a soil sample. It's from an area that I just put IMO actually about a month ago. So it's good to see out there. Okay, so can you see that thing up at the top? What is that?
Leg.
Another fungus.
This thing that's twisted right here?
Oh, it's twisting. I
Vegetation.
can't see it.
So you can see how I zoom through it and you can see how it twists. So you see that it's not a fungus because it's twisting like that. So I know it's not. And so what that is most likely is some sort of plant material, some sort of like fiber, some sort of other deal in there. So I think that's another chunk of fungus there. Maybe, maybe not. Sometimes it's hard to tell, but I believe that is. And so here, okay, yeah, so here, so this guy here, this is a fungus growing throughout my sample, but is this one beneficial? This guy right there.
It looks dark.
But I would say because this guy is much thinner, that he's only about 2 micrometers across, I would say that this chunk is most likely not actually a beneficial fungus. Even though he is of the dark color, the size being making more of a difference, and then the color being the secondary part of that.
If it's not beneficial, is it necessarily detrimental?
No, it might just be like benign, like just hanging out.
Okay.
Then there's this here, which this is either either an algae or a piece of pollen. Both algae and like little diatom type of things look really fractally, and so do pieces of pollen that you'll see. So chunks of things like that in there are pollen or an algae, depending. These big clumps out here, these are what you would call, when you're looking at this sample, you would call this is an aggregate. When something is stuck and clumped together like this is an aggregate. So this is just a bunch of bacteria glued together on some particles of dirt or in some other minerals and things in the soil that are glued together. And if you need to count bacteria, for some reason, which I don't do that, but you would have to just dilute this more and then see, to break these aggregates up to see, because this is probably like, I don't know, thousands of them. I can't make a good estimate on that. And that's one of the reasons when I'm doing my soil analysis, I largely ignore the bacteria. Because if I have good fungus, the good fungus will keep the bacteria in order, and the bacteria that arrives tends to be beneficial as well. If I have beneficial fungus, I guarantee I have beneficial bacteria. So just looking for the fungus, which are easy to see, easy to identify. I don't have to worry about DNA sampling these guys to find out who they are. I know if this guy is here and he's beneficial, that they're good guys and they're on his team. They're hanging out together. Yes?
What's the mechanism by which they regulate the bacteria?
Different things that they'll secrete, different hormones and enzymes that they secrete. Even like the plant itself will emit different sugars to change the bacteria and the fungus in the soil. And then the fungus itself can put out certain antibiotics or probiotics to also change that relationship. So on here you might see all these tiny little things and think there's a lot of life and I have a lot of active bacteria. But really what you're seeing is just the water and the slide, and just because the fan's on the table, it's making it vibrate. It's just vibration of Brownian motion is what they call it. And so these are not active and moving, even though they look like they're jiggling around. To see an active and moving one, it wouldn't be just jiggling, it would actually be like an intent. Like you'd see it move down and then like across, and then, you know, like it'd be on a pattern, not just jiggling. These are jiggling. So yeah, I haven't seen any active stuff on here yet, or any larger life, but good fungus here so far. And I'll go to one last random spot, and then so I'm like tracing this fungus or whatever it is, and if that is a fungus, it's definitely not a good one because it's so skinny and thin, and it's also up like that, and so I'm tracing through to see it. But sometimes you have to change how you're focusing to see what's in every slide. But yeah, check this out for in terms of like fungus on my slide. Like, so I got some good stuff going here. And another thing, you see how this guy has segments in him?
Mm-hmm.
This is different than the yeast that were stuck together. This is actually one fungus with these segments. And the segment puts them in a special family of fungus, but these tend to be also beneficial when they're segmented like that. Cool, so that was that sample. I gotta clean up some equipment to like do the next sample. But how do you want to do it? Do you want to all continue to look at these samples and go through the IMO?
Sure.
Yeah, I like seeing them up
I
there
like
when
seeing
you can
that
explain
way.
it to us.
Okay, so I'll put the IMO up here. Like, yeah, can you rinse that out?
Yeah. Anything else you need to clean up?
Yeah, actually I brought a bunch with me, but no, I'm good
It's
on
just
that.
one.
Slide and be clean, but I got another one. Hundreds of layers. I recommend recommend you try to get glass slides too, if you can, even though they're fragile and it's sometimes they get stuck in your fingers. Plastic is not as easy to work with.
You use the slides, have a little depression in them?
No, I don't. I do have one. There's one like this one right here has the dimple in it.
Yeah.
But I'm trying to kind of get it flat and not have too much depth. Because like the organisms are kind of going to die. I'm going to smush them.
You done with that slide?
Yeah. Looking for a clean copy slip that's not shattered.
Or you can lower it, so, yeah, just so there's more space.
Cool, and so I didn't mention, but I learned this slide stuff from Elaine Ingham, who's famous for the Soil Food Web. It's her company. And she taught this microscope course in 2012 in Kohala, and we learned all about composting and all about this biology. And I thought it was amazing because the natural farming stuff was like the optimal already. Like, and she was going through composting to kind of get to this process, but natural farming was the optimal. And this gave me the tools to analyze the natural farming. Because before that, I'd been like, you make up an IMO 4 pile, or you make your IMOs, and like, is it good? Is it not? And so this tool right here, and understanding the beneficial fungus, gives you a real quick. You can save time, money, energy, all that, just quickly. And especially when I'm doing compost teas and brewing things, I like to look at the organisms before I use them. Doing an experiment with my mom right now on flowers, and you know how the waters super stink after like a week of flowers, and it smells just horrible. We found that if we put the lactic acid bacteria in there, there's no smell. After the 2 weeks, you pull it out, and we verified that with the microscope, that the untreated batch, untreated batch just had all these snakes in it. These tiny little, like, mosquito larva looking things. And that's what's stinking. Like, your body is telling you, do not eat those guys. All right, and so this is the IMO. The IMO is sometimes more exciting if you soak it in water a little bit first, and you kind of activate it, because sometimes it tends to be too dehydrated. But this pile was outside already still going, so I just pulled it where it I think it had enough moisture, is what I'm trying to say.
Is there a place on the web you can look for identification of soil bacteria and funguses?
Sort of, not really. Like, it's hard to find the right level of, like, some are super scientific, and it's hard to find ones that are like practical.
Yeah.
That I found that taught me, like, what I need to know. But I do think that I was talking to Vivian, who took Elaine's course as well, and she was talking about making like a online database of where, because I can take pictures with the same camera.
Yeah.
And then just like having a database of it. This is what we found in this area, because just in our little conversation, I found out that she has never seen a flagellate, and maybe she didn't know exactly what it was or whatever, but she'd never found one in all her samples, but she finds amoebas all the time. And I look at my samples, and I've seen a fair amount of flagellates, not a whole lot, but I've seen them, but I've never seen an amoeba. So, and she lives up in Canada. She's in like, I don't know, where they, French Canada, like the other side. And so just like, that's just an amazing, like, you know, like if we're sampling our yard, like she's naturally rich in amoebas and we're naturally not. Like,
Interesting.
so yeah, having a database of soil, I'm gonna put 2 drops on you. Okay, so first thing first on like when you get a new slide and you put it in is that you can scan through, which I kind of reduced the light here for me to see anything. Can you still see it there? So first thing first is I scan the slide just to look for nematodes at 40 times. Because you can see them, they're big enough that they're riding around that it's worth a little bit of time to immediately, just before you start to really look into the slide, to just go across. And this is the edge of my slide. You guys see that? That's the edge, so I can see from edge to edge, just kind of cruising across my slide. See if I see any nematodes.
What are these round circles?
Oh, these. So,
Air bubbles.
yeah, no, like, yeah, there's lots of stories about them, but they're air bubbles.
Oh, okay.
And sometimes they look
pretty
fantastic.
cool.
You're looking at it, you're like, whoa. But it's just air.
Come to close the chickens.
Okay.
Okay, and so I did not see any nematodes. I scrolled the whole thing. I was hoping to find one, because it's always nice to see and share. That might be a beard hair.
They're everywhere.
Yeah, right. All my samples in there. Okay, so I scrolled through it 40 times and find any nematodes. So I'm just gonna zoom into 400 times, and 400 times is what I'm using to look for my beneficial fungus. Get more light.
Do you expect to see nematodes in the IMO?
Yeah, because it's such a rich environment that they're in there.
What is a nematode?
A nematode's like a small snake, and interesting fact from Elaine is that they took the nematodes and put it in a jar with no food, and then left it for like a decade or longer, and came back and put it under the microscope, and they're still moving around.
Oh, wow.
That apparently they don't need to eat food to live. They just do it 'cause
Or we
it's
don't know what they're
delicious.
eating.
Living on photons.
So this is the IMO sample, and not nearly as exciting as the soil sample, I don't think, at least. But do you notice all these round things that I'm seeing? All these round things in here. What those are typically are fungal spores or spores of some sort, because this IMO is dry and then they're in their hibernation form. So it's as if they're like in an egg, they're in their hardened off state. And that's why I said that if you moisten the IMO for a while and then you look at it, these will hatch, and then it'll be much more exciting to see. But right now they're just kind of sleeping in here. A great way to wake them up is by adding worm tea, which has lots of humic acid in it, and it really wakes the fungus up and these guys up. So yeah, tons of spores. You see, it's just like kind of covered with spores. I did see some interesting things. So it's hard to say, like, looking at your soil to judge when you're at the equal parts of fungus is easier versus looking at your IMO. Right? So, glad I started with the exciting one. Any further questions on this?
Is there a way to tell that your IMO is got the right proportions of what you want it to have? Do you know that you got a good batch?
Yeah, when you're, like, if you put out your collection, your simple starch, before you add the equal parts of sugar to preserve it, you can take that and, same like I did, just shake that up and then see what you get.
Okay.
Yeah. Yeah. As soon as you add the sugar, they're gonna not be active, and you're not gonna see them.
And what you want to see in a good IMO like that would be equal amounts of bacteria and fungi?
Equal by weight. It's not equal amounts.
Weight, yeah.
The
Okay.
bacteria are gonna far outnumber the fungus always, but by weight.
Okay.
Which I'm just, yeah. So I'm just estimating the biomass too. You know, like based on, if it's greater than 3.5 micrometers and I see this many, then that gives me an estimate of the biomass.
So that was one per every view. If you saw one fungus per every view, then you would have equal parts, you think? Okay.
Yeah. And also, like you saw, I was cruising around, I saw predominantly beneficial fungus as well. And so that's another thing. If I'm seeing predominantly not beneficial fungus, then I know, well, you should make actually actinobacteria to go in and they kind of clean the slate for you.
When you say one fungus per view, what's your magnification?
400 times.
400.
Yep. Yeah, I think those are all good questions that I kind of like. It's like a technicality.
How many kinds of fungus are there?
Lots?
Two.
They're trillions. In this bag alone, there's like a trillion, right? And so and one of the best things about this method is that the diversity is so. That's what we're trying to encourage. So when you're mixing your pile, even if you take the microbes from here and you mix it over there, you're now creating this diversity. It's like mixing the universe together to get all these different things. So, thousands. They scientifically, they don't even know. Like, they estimate they know, 2 to 10% of all that exists. Certain ones you cannot grow. The reason I use this method here, certain ones you cannot grow plating. So in microbiology, you take the sample, you put it on a plate of stuff, and it grows, right? When you put the lid on top and you cut off the oxygen, only stuff without oxygen is going to grow, and only stuff that grows on that's going to grow. So with this microscope, you can see stuff that grows in the soil and stuff that grows with oxygen. And so it's using this live method here versus trying to plate to see what's going to grow gives you much more of an idea what's happening in the soil. Most of the microorganisms we know about are the ones that grow on the plate because those are easy to grow and those tend to actually be bad guys that attack you. And so we know a lot about them, but we don't know a lot about the good guys that don't grow in the, they grow in the soil because they're so diverse and because they're so amazing, but we can look at them. Any other questions?
Are you gonna do your other sample or no?
I can and I will. I'd rather, I guess, I brought some food. Eat some snacks and then, yeah, we can check things out.
How do you increase your fungus count in the soil?
Collecting where you know there's good fungus already. So then feeding it the natural farming inputs, which tends to really increase the biology, the beneficial biology.
Okay.
Any other questions?
Thank you.